Assalamualaikum warahmatullahi wabarakatuh. Today, my teammates and I want to make a video presentation for subject AGR506, which is Agriculture Biotechnology, and the topic that we choose is from the chapter 5, which is Biological Control Agents. First of all, please allow me to introduce myself and my teammates. I am as a first presenter. My name is Muhammad AZRI bin ZAINUDDIN, and my metric number is 2023399653. Next, the second presenter is LATIF NURHAFIZ bin HASARUDIN, and his battery number is 2023502041. And we are from the same groups, which is M3AT2223A. Firstly, we jump into the first part, which is types of biocontrol agents. There are two types of biocontrol agents, which are inductive biocontrol, which is uses pathogens to target wheat at a very high rate like herbicide application. Most common pathogens include nematodes and gnats. This approach does not prevent the invasive plant from implementing at a later date. Next is a classical biocontrol, which is natural predators of the invasive plant to create an eternal relationship between a plant and biological control animals. Next, we move to an antagonism mechanism. In phytopathology, antagonism refers to the action of any organism that suppresses or interferes with the normal growth and activity of a plant pathogen, such as the main parts of bacteria or fungi. These organisms can be used for pest control and are referred to as biological control agents. There are several antagonism mechanisms in biocontrol agents. Firstly, commensalism, competition, predator and parasitisms, and lastly is a cytophore. So we go for more into it. Commensalism is one population affects the growth of another population whilst itself being unaffected. Metabolite penetrates the cell wall and inhibits its activity by chemical toxicity. Other plant residues are the source of phenolic and non-volatile compounds. Antimicrobial substance, which is antibiotics produced by microorganisms in soy bacteria, actinomycetes, which is a fungi, aldehydes, alcohols, acetone, organic acids, non-volatile and volatile compounds, which are toxic to microbes. Change in microbial structure, which is cell wall, hyphae, conidia, and the others, may occur when microorganisms lack resistance against the attack by deleterious agents or unfavorable nutritional conditions. Next, we move to the cytophores, which is the other extracellular metabolites, which are secreted by bacteria, actinomycetes, yeast, fungi, and dinoflagellates. Microbial ion chelating compounds because they have a very high chelating affinity for iron 3+ ions and a very low affinity with iron 2+ ions. After chelating iron 3+, they transport it into the cells. After chelating iron 3+, it makes the soils iron 3+ deficient for other microorganisms. Consequently, growth of other microorganisms is inhibited. It supplies iron to plants, and therefore plant growth is stimulated. The other parts of antagonism mechanism is competition. Rapid germination of fungal propagules and fast growth of young hyphae towards a source of soluble nutrients. Appropriate enzyme equipment for degradation of carbon constituents of plant tissues. Excretion of fungistatic and bacteriostatic growth products including antibiotics, and tolerance of fungistatic substance produced by competitive microorganisms. Next, predation and parasitisms, which contains three elements. Firstly, mycoparasitism, The term microparasitism applies strictly to those relationships in which one living fungus acts as a nutrient source for another, but fungi-colous relationships may also be included in which nutrient exchange has not been shown. Next, nematophagy. Nematophagous fungi are carnivorous fungi specialized in trapping and digesting nematodes. Species exist that live inside the nematodes from the beginning, and others that catch them, mostly with glue traps or in rings, some of which constrict on contact. And then lastly is mycophagy. Mycophagy have three section, which is from A to C, that to feeding of amoeba, open fungi conidia, and a fungi hypha. Firstly, there are attachment of amoeba, and secondly, the engulfment and hole formation, and the lastly is a digestion of conidium. Next one, we move to strategies of biological control. Firstly, importation. One of the earliest success in West was in controlling Escherichia purchasei, which is a cottony cushion scale. In Australia, they are using a predatory insect Rhodolia cardinalis, which is a Vadalia beetle. Secondly, the augmentation. Augmentation is a supplemental release of natural enemies, boosting the naturally occurring population. It is release additional numbers of a natural enemy into a system or modifying the environment to promote greater number of or effectiveness. Lastly, the other strategies of biological control is conservation. Conservation is avoidance of measures that destroy natural enemies and the use of measures that increase their longevity and reproduction of the natural enemies in an environment. Natural enemies are already adapted to the habitat and to the target pest, and their conservation can be simple and cost effective. For the next slide, I will allow the LATIF to continue for biological control agents.
There are three main components in biological control agent, which is predators, entomorphogenic parasitoids, and entomorphogenic microorganisms, which comprise of bacteria, fungi, nematodes, and viruses. Firstly, I will explain about the predators. Predation is a biological interaction where one organism, the predator, kills and eats another organism, the prey. It is one of the most common interactions that exists in the nature that serve as feeding behavior for living things in the world. Other than predation, there also exists micropredation, such as parasitism, which usually do not kill the host, and parasitoidism, which will usually kill the host but will take a long time. Example for predation is ladybugs and spider that preys on aphids that are destroying the plant. Next, I will explain about parasitoids. Entomorphogenic parasitoids agent will usually lay their eggs on or inside the body of an insect host, where they will serve as protection and feed for the parasitoid's larvae. The host will be ultimately killed when the larvae is fully matured. Most common entomorphogenic parasitoids used in agriculture is Ichneumoniid wasp, which prey mainly on caterpillars of butterflies, and Baraconiid wasps, where they attack caterpillars of wide range and other insects including green fly. Thirdly, there are bacteria. Bacteria used for biological control infect insects through their digestive tract, thus preventing them from feeding on the plant leaves and fruit. However, they have some limited options for controlling insects, where only sucking mouthpart insects such as aphid and scale insects are infected. For example, Bacillus thuringiensis is most widely applied species of bacteria used for biological control, and it can be used against Lepidoptera, Coleoptera, and Diptera. In addition, there are also fungi. Fungi protect crops from a variety of pests. They can infect a wide range of insect hosts. However, there has been only limited success in fungi in biological control agent, as fungi are slow in killing their hosts. The fungi mode of action involve addition of the spores to the host cuticle, spores germination, penetration of the cuticle, growth in the hemocytes, death and saprophytic feeding, and hyphal reemergence and sporulation. Moving on, we have nematodes. Nematodes are microscopic roundworms used in biological control to manage agricultural pests. They infect and kill insects, reducing the needs for chemical pesticides. They are environmentally friendly and target specific. Thus, nematodes help in maintaining ecological balance while protecting crops. Examples for nematodes are Sternema carpocapsae and Sternema affinis. Next, we have protozoa. Protozoa are single-cell organisms used in biological control to manage agricultural pests and disease. They can parasitize or prey on harmful insects and pathogens, thus enhancing soil health and crop protection. Protozoa offer a sustainable alternative to chemical treatments and promoting a balanced ecosystem in agriculture. For example, Malamoeba leucostae, mainly known as a contaminant of laboratory cultured locusts. Finally, we have viruses. Viruses are obligate disease-causing organisms that can reproduce within a host insect. They provide a safe, effective, and sustainable control of variety type of insect pests and usually attack larvae of Lepidoptera or Hymenoptera. Viruses are host-specific and induce mortality by accumulating toxic protein during reproductive cycle of virus after death of insect, causing the integument and internal tissue of the insect to melt away. For the next slide, I will explain about the advantages and disadvantages in using biological control agent. Firstly, the advantages are they are environmentally friendly and cause no side effect. They cost less compared to pesticide and insecticide and easy to use and is effective throughout the season. They also help in reducing the use of chemical and other pesticides. Meanwhile, the disadvantages are it affects the product quality and pest is not completely destroyed. It also only effective for large scale production and they also takes a lot of time to take effect. And thus that's conclude our presentation. Thank you.